genetics, principles of inheritance and variation that we are going to see today, mutation. In the last class, I have explained to people about the sex determination, isn't it? I hope you are clear with that. So let me explain to people today about the mutations, right? Mutations means it is a, I can say if I use a single word, I can say that it is a change. But actually, what mutation we can say in the definition that is a spontaneous permanent change in the genetic material of an individual that is called as mutations. Right? So, one more time, I would like to repeat the definition and want to explain you what exactly mean by mutation. Mutation means change. Change means where? In the genetic material. What is the genetic material? Genetic material is nothing but DNA. Okay, genetic material is nothing but DNA, isn't it? So, there is a change in the genetic material means that we change in the so many things that we are going to see in the mutation. Okay, the mutation topic is spontaneous, permanent change in the genetic material in the DNA of an individual that is called as mutation. Was it okay? According to a scientist called Robson, what he said that mutation in a single word, we can say that it is a mistake or misprint in cell division. What it is mutation? Mutation is a mistake or misprint in cell division. What exactly is happening in the cell division here, students? During the cell division, the nucleus is having the chromosome. The chromosome consisting of DNA, isn't it? So these DNA, that is a genetic material, has to be doubled during the process of cell division. Why? Because a same copy we should get, that is two same copies we want to get. So the material should be doubled before it is copied, isn't it? So that is the reason when it is copying, there is a mistake. If there is a mistake or misprint, then what happens? Change in the genetic material. For example, if you are reading something, if you have something what we call it as some sentences in your book, if you are keeping instead of your book, you have taken somebody's book. So some changes might be there, some grammatical changes, something. So I can say that there is some mistake or misprints. So which is making the difference between your copy and their copy. Isn't it? If you compare your copy, suppose if yours is an original one. So when the next one is, your friend is copying by using your copy, so he or she is doing some mistakes in it. So that I can consider as a mutation in case of DNA. That is a mistake or misprint in the cell division. That is during the process of cell division, the genetic material is doubled. When it is doubling, means how it is doubling? Suppose I have this marker pen. Okay, I want one more. So what I am doing, I am copying. I am trying to make same similar like this. So if I do some mistake, so this original will be differing from the newly made one. So now there will be difference, isn't it? I can call it as a mutation. Yes, it is a mutation. It means that it is a change. How I got this change? Because of my mistake or because there is a misprint or some of the other way. Okay, that we call it as mutation. What results in the mutation in case of the genetics? So you know that in the molecular basis of inheritance, you have studied that the DNA you have a very tiny cell in that very tiny nucleus. That nucleus is packaging a long, that is meter of DNA is being packaged in that consisting of so many nucleotides. If there is a mistake, suppose if the DNA is having that is the sequence is A, C, G. So continuously like this it is having. Okay. So if there is a mistake here, instead of G, if it is C once again. So there is a mistake here or misprint here. Because of that reason, the original DNA will be differing from this DNA. That I can call it as mutation. Understood about mutation? Okay, my dear students. What is the reason behind why this mistake is taking place? So I will tell you the reason what exactly the mistake is. Okay, so if you see the reason why this mistake is taking place, isn't it? So during the process of cell division, it is either because of the DNA duplication, some mistake will be there, or it is because of the chemical or some of the other radiations. This also causes mutations. Change in the genetic material. You know that how sometimes cancers are causing normal gene is due to the radiation, UV radiation. If we come in contact with the UV radiations, what is going to happen? Normal cell is going to be mutated into carcinogenic cell. So at that time, that cell is capable of producing cancer, isn't it? So mutation is taking place. So what is the reason? Reason for that, isn't it? Why? If I say mutation is taking place, so you can ask me why, madam? Why? What is the reason? The reason may be because of the DNA duplication, or it is because of the some of the chemicals also. Some of the chemicals also we can see there is a mutations. Chemicals also have the capacity to change the mutation. Mutation in the sense we can also say that the nucleotides will be there in the adenosine, guanine, cytosine, thymine, these all will be binding together. So when they are forming the nucleotides, there is a sequence, one will be missed or changed. I'll tell you in the types of mutations. It is point mutation and that is gene mutation and chromosome mutation. I will be telling you people. So the reason maybe it is because of the DNA duplication time. Okay, or it is because of the some chemicals which are capable of doing mutations, and also because of the radiation. Some of the radiations also are causing as a mutations in the genetic material. The substances which are causing the mutations, we can call it as mutagens. What they are called as beta? Mutagens. The substances which are capable of forming the mutations, such substances are called as mutagens. For example, chemicals, some of the chemicals, radiations like UV radiations and all, they will be causing mutations in the cell, isn't it? So the substances which are causing mutations are called as mutagens. You people might be in the might be knowing that mutations will be asked for 2 or 3 mark questions, isn't it? It will also ask in 1 mark mask also. In 1 marks, very rare question. If they will ask in 1 marks, they will ask you the definition of mutation and definition of the both the point mutation and that is gene mutation and chromosomal mutation. If they ask in 2 marks, what is the possibility they are going to ask you for? They'll be asking what is mutation, then you have to write the definition of the mutation and write the 2 different types of mutations. You have to just write the 2 names. But sometimes they will be asking the question in 3 marks, definitions of mutation and what we call it as you have to write the types of mutation definitions also, isn't it? Types of definition, mutation, also they will be asking, isn't it? So that will be for the 3 mark question. Now let us talk about what are the different types of mutations. The different types of mutations we can see here, that is gene mutation and chromosomal mutation. Gene mutation and chromosomal mutation. Gene mutation is also called as point mutation. Point mutation. We can call it as point mutation. gene mutation and chromosomal mutation. Gene mutation is also said to be point mutation. Okay. So whatever the mutations if you are seeing, you know that in our body there are two types of cells, somatic cells and gammatic cells. Somatic cells, gammatic cells. Somatic cells which are helping to repair our body, isn't it? So whatever the changes taking place in the somatic cells will not transfer to the next generation. Will not transfer to the next generation, isn't it? Keep it in your mind. Okay, dear students, if the changes is taking place in the gammatic cells are nothing but gametes. It may be in the sperms or it may be in the ovoids, because the fusion of this two only will be resulting in the formation of a zygote. The zygote will develop into the embryo, then embryo will develop into the egg one, isn't it? So if there is a change in the gametic cells, there is going to be changes in the, it is going to be transferred to the next generation. If there is a mutation here, there is no changes almost, but here there will be changing. So here I am talking about the gene mutation or point mutation. If there is a mutation among the, what is a gene? It is a sequence of DNA. Gene is nothing but it is a sequence of DNA. So you can see change is conformed only to the gene. The resultant chain sequences of arrangement of the nucleotides. For example, DNA molecule, example, hemoglobin is a protein containing 300 amino acids. Hemoglobin, you find hemoglobin in the blood, isn't it? That is also secreted, that is also produced by the every gene, every characteristics of our body is responsible of the expression of the gene. What is gene? Gene is nothing but a segment of DNA. A segment of DNA. So here, if you see, there is a changes. That is a hemoglobin, which is a protein containing 300 amino acids. Depends upon a specific gene. If there is a change in one atomic acid, if it is replaced by valine, at that time it is going to cause some of the disease called sickle cell anemia. Anemia, you know, anemia. It is the cell RBC cell shape will be changing. What is going to affect at that time? So oxygen carrying capacity will be lost. It will be changing, isn't it? That anemic condition. Sickle, the RBCs will become sickle shape. I would like to explain you one more time here. Suppose okay, so like this it will be a DNA sequence. It will be one. Understand how does the DNA is understood beta? So this is a sequence, for example. So each amino acid is made up of by coding of 3 nucleotides. Suppose this is coding for one, this is coding for another. So likewise, in the formation of the hemoglobin is a type of protein. All proteins is made up of amino acids. So this hemoglobin is made up of how many amino acids? 300 amino acids it is made up of. How many? It is made up of 300 types of amino acids it is made up of. Okay, 300 types of, sorry, not types, 300 amino acids it is made up of, isn't it? So these 300 amino acids arranged in a sequence. For example, 1 to 300 when you are writing 1 to 300 you are writing the sequence, isn't it? So when you are writing in the sequence, so what happens? For example, when you are writing or you have written in a directly file. So then you write completely correct. Then also what happens, my dear students? Then also the sequence will be changing. Okay, instead of 4 you have written 5, 1 only you have missed. But if even though you write continuously it will be 100 character, but it is changing. Only because of one, that is called as point mutation. Point mutation. One more will be the frame shift mutation. Complete one nucleotide is getting replaced by another one. Okay. So likewise, here what happens beta? That is the mutation is going to occur at a particular point, that is in the gene sequence, that is in the DNA sequence. That is what we call it as what beta? That is gene mutation or point mutation. We can call it as gene mutation or point mutation. We can call it as. Okay. So example. Suppose this is coding. This is coding for one amino acid. This is coding for the another amino acid. So the sequence which is coding for the glutamic acid is changed. So it is changed at the time what happens? It is coding for the valine. That sequence is going to be replaced by this one. So instead of glutamic acid, in the beta chain of the synthesizing hemoglobin synthesizing beta chain will be there now. So that chain is being replaced by some other sequence. So instead of glutamic acid, it is replaced by valine. So at that time, the normal RBCs will not be there. They are changing into sickle shape. So we call it, this is that is called as sickle cell anemia. It is one of the types of genetic disorder. It is inborn genetic disorder. It is okay. So this is said to be genetic disorder now. So what about the chromosomal mutation? Here the mutations are directly taking place in the chromosomal level. If, for example, change conform to the chromosome. Okay. For example, I said here the changes conform to the changes are going to be conformed to genes. यानी कि only gene level पे changes आ जाते हैं, यानी कि sequence of the DNA में change आ जाता है, isn't it? But यहाँ पे क्या होता है? The changes taking place conform changes conformed to chromosomes. Okay, dear students, changes are conformed to the chromosomes. What happens? I have said you changes conformed to the gene. So gene के sequence में change हो रहा है. यहाँ पे chromosomal mutation में क्या हो रहा है? Changes conformed to the chromosomal level. Either the, for example, 23 pairs of chromosomes we find in the human beings. My dear students, if you see the human beings chromosomes, you can find that only 23 pairs of chromosomes are there. As well also I said, it is very clear, 22 pairs of autosomes. Autosomes will be there. Okay, they are responsible for our body character. So they are called as somatic chromosomes. Okay, students, the one pair, that is 23rd pair, that is only one pair, is responsible for, that is called as androsome. Here, this pair is responsible for determining the sex of the organism. So it is called as sex chromosome. Isn't it clear with this data? Because only 23 pairs are there. If for the these 23 pairs, can you be writing in the sex determinant in the disorders are telling you that how it will be. Among these 23 pairs, what happens? That is 23 autosome plus 1 allosome, isn't it? So here it will be there 22 autosomes here, for example, along with this pedigree, here 1 plus will be there, extra will be there. So it is increasing, that is change, mutation. The exact number will not come. Now how it will be? 23 plus 1 means 24, which is not normal. Which is not normal. Okay, abnormal. So either increase, addition of the chromosome or deletion of the chromosome will affect the normal number, that is 23 pairs of chromosomes. So I can call it as mutation, chromosomal mutation. I can call it as the students. Exactly, I can call it as there is a change in the chromosome. I'll show you here. 100% there will be change. Either due to decrease or increase in the, that is addition of extra chromosome, or removal of one more chromosome will be disturbing the normal number of the chromosome. That is called as chromosomal mutation or chromosome mutation, isn't it? So we can call it as chromosome mutation, isn't it? I can show you here. So 23 pairs, total 23 pairs. Among the 22, autosomes will be there. Autosomes may associate the 21st pair. 21st pair. For example, this is first pair. Okay, first pair. Second pair. Okay, third pair. Okay, SLE. If you see, for example, 21st pair is वहाँ पे क्या हो गया? Instead of 2 chromosome, 1 added. So this is, we are adding 22 chromosome plus 1, that is equal to 23 plus 1 sex chromosome, that is allosome. So what will be the total karyotype? Total will be 24 pairs. Is it normal? No, it is a mutation. Why it is? It is because of the in genetic disorder, chromosomal disorders. I will be showing you people. After this, I will be telling you the chromosomal disorders. In that, I will be explaining you, dear students, about this. Okay, so it is not normal. It is not normal. It is abnormal. For example, in this one pair, normally I said it is now. So for example, what happens now here? Addition, I have shown you people. For example, that is allosome, that is a sex chromosome. For example, 22 pairs have been allosomes, which is correct. Autosomes, correct. Then the one pair, that is instead of XY, one more X is present. Then what happens? Addition. That is due to addition, again there is a change. Again instead of 23 and 46 chromosome की वजह से क्या हो जाता है? 47 chromosome हो जाते हैं. Again it is an abnormal chromosomal disorders in the study. Now let us talk about the pedigree. Pedigree analysis. What do you mean by pedigree? Pedigree analysis. Have you people, if I ask you people, can you tell me anybody in your, if your parents, they are showing curly hairs, your parent, father is also having silky hairs, mother is also having silky hair, even though the child is showing curly hair, or cat eyes, you know, even though the parents are not showing any type of cat eyes, but your grandparents, either your maternal or your paternal, मम्मी के साइड वाले जो नानी नाना होते हैं या फिर पापा के साइड वाले जो दादी दादा होते हैं, उनके साइड में अगर कैट आइज है, इवन तो आपके मम्मी पापा में नहीं है, आपके भाई बहन में या आपको कैट आइज दिखाई दे सकते हैं. So if I ask you one question, can you tell me in your ancestor till today, what is the possibility to how many of them they had the cat eyes, curly hairs, or six fingers? We have pentadactylus. If one extra finger is present, like Hrithik Roshan, what happens? Is it possible everybody will be having? No, it is because of one change, isn't it? So how can we analyze? We can analyze by using, that is, pedigree. That is what we say, that it is a pedigree analysis. Family tree we can pedigree करके हम लोग देख सकते हैं यहाँ पे. Pedigree is in actually few more exam only for 1 or 2 marks, not more than that. Okay, for 1 mark only definition, if it is asked for 2 marks, they will be asking you to recognize the symbols. But in need, one crossing autosomal recessive trait or dominating trait related one question will be asked for you. Definite question will be there in the NEET and also in the CET part. So, what exactly do you mean by pedigree analysis? Pedigree analysis is nothing but it is a record. It is a record. What we are going to do is, it is a record of inheritance of traits in several generations of a family. Okay, it is a record of. It is a record of inheritance of traits in a family, in a family, traits in a family generation from generation to generation. Okay. So, if you see, for example, the trait may be, what may be the traits? The trait may be eye color. The traits may be texture of hair, eye color, skin color, certain genetic orders. So the traits may be, what may be the traits? The traits may be texture of hair, eye color, okay, skin color. It not only the diseases also will transfer. That also we can say that is genetic disorders. The diseases which are transferring from the parents to the offspring or from the grandparents to the. It doesn't mean that if your parents are having, none of them should have. If your grandparents are also having, sometimes the genes will skipping genes will be there. They won't appear in the next generation. They will be appearing in this generation. So the diseases which are transferring from the generation to the generation, that is from the parents to the offspring, we call it as genetic disorders. I will teach you. After this topic, I will explain you that only to the students. So that also we can analyze. We can analyze that also कि फैमिली में पहले ये डिसीज किसको था, दादा को था, नाना को था, या दादी को था, या नानी को था, या फिर आपके चाचा को है, या मासी को है, पापा को है, मम्मी को है, फिर फ्यूचर में किस को हो सकता है. That also we can analyze this through the pedigree. Okay, so one more time I explain to dear students what exactly pedigree is. Pedigree analysis is nothing but it is a record of inheritance of traits in a family from generation to the generation. Pedigree चार्ट को देख के आप एनालाइज कर सकते हैं. जो जेनेटिक काउंसलर रहते हैं ना, जेनेटिक काउंसलर्स रहते हैं. काउंसलर मीन्स for example, normal counselor will counsel like you should be like this, means to sort out your problem, whatever the problem is, isn't it? You will go for the counselor, isn't it? So likewise, genetic counselors will be there. So those will be analyzing this pedigree chart and they will be recognizing where exactly the disease was and now what are the possibilities in the next generation. कुछ क्या बोलते हैं कुछ लोगों में कुछ जेनेटिक डिसऑर्डर्स रहते हैं फ्यूचर में चिल्ड्रंस में आते हैं क्या देखने के लिए जेनेटिक काउंसलर्स को वो दिखा सकते हैं पेडिग्री से जेनेटिक काउंसलर्स कैन स्टडी एंड एनालाइज कि पहले किसको था अब किसको है और फ्यूचर में किस किसको आ सकता है सो इट इज अ रिकॉर्ड ऑफ इनहेरिटेंस ऑफ traits only na hair color texture of hair some will be having silky hairs some will be having very soft hairs some will be showing rough hairs some will be showing curl hairs isn't it so what about the eye color eye color will be normal eye color we are showing some people will be having brown eyes like cat eyes and very rarely blue eyes like Aishwarya Rai isn't it that is difference it may be inherited to the next generation. It might skip, it may inherit later on. Okay, so where it was there, and by seeing that pedigree and a chart, a person can analyze कि ये trait कहाँ से आया है और next generation में कहाँ जाएगा. Even though skin color also we can say. We have the varieties, isn't it? The skin color varying. For example, if you go to African grows completely black, Africans you know that. It is because of what we call it as directly the sun will be there, melanin synthesis will be more in the pigmentation is more than it is showing very darkness, isn't it? The same, for example, if you go to the American countries, you will be having both white and black. The white melanin pigment only will not be there. So they are showing completely white. In our India, if you see, you find greenish color, okay, dark color, completely fair, pinkish color, different skin color. That is also responsible because of the expression of the trait. Isn't it? So now genetic disorders, sickle cell anemia, phenylketonuria, the genetic disorders which we are going to study, which we are going to be discussed in our, I hope today or any next class. If it is possible, today only I have started, or else we will continue the next class. So the diseases which are present inherited from one generation to the another generation, that is called genetic disorders. Okay, that also we can analyze because of this pedigree. So now come to the another important point. How can the person be analyzed by seeing the chart? There is some symbols. Symbol is there. For female, symbol is there. Affected male, unaffected male. Sorry, infected male, infected female. Okay, beta. So now I will show you how can we recognize. There are some symbols. That is this symbol. Each symbol is representing for one or the another things, isn't it? So this is recognizing the male, and this is the female, and this is unspecified sex, neither male nor female. There are some persons neither showing maleness nor femaleness. We are representing the symbol, and if it is then there is a line, what we call it as it is joining these two, that is a marriage or mating between individual. Okay, then at the symbol, for example, these for normal male, normal female. This is unspecified sex. If it is these symbols show that they are affected or affected by affected male. Okay. Then affected female. Okay, dear students, this will be the unspecified sex. It is affected unspecified sex. If you want to see, this is the parental generation. It is a marriage between the male and female. If I will write the symbol between like this, marriage between relatives, marriage between the relatives. Have you seen? Usually we say that uncle's son or one of the nearby auntie's daughter. We are making the marriages between the relatives. So at that time, it is going to affect. Continue marriages between within the same relatives may cause infertility, and also there are some more other chances in this study later on. So whatever is related here, so I can say you people that is mating between the relatives. Now here, if the symbol is showing, okay, then. Can you tell me what it indicates? It indicates parents with their next generation. Parents with their next generation. For example, if I do like this, what it indicates? These are the normal female, affected male, normal male. If I indicate like this, unspecified sex. Isn't it? So, I go to right here. So, it is showing that is above parents and below offsprings. Okay. So again, the same things will be following. If it is affected, we can draw a line between that. So one more time, I can tell you people how the symbols are indicating. The symbols will be like and for example, directly also we can show. This is unspecified. How many among these children are unspecified? 6, 5. I directly can show 5 affected. Okay, some books, but these are very clearly shown. Commonly used symbols are these many what I have written on the board. Okay, so once again I would like to explain what are the symbols we are using in pedigree. Squares represents male sex. Okay, in the pedigree the squares represents the male, whereas the circle indicating the normal female, and here it is unspecified sex, neither male nor female. Then, if there is a line between this, so vertical line, horizontal line, you are able to see that is marriages between the male and the female. You can also call it as mating between the male and the female. If there is a two lines, it indicates that the marriages are taking place between the relatives. So that if there is so many lines indicating the male is affected, and so many lines indicating the female is affected. Same unspecified sex, and that is indicating parents above and the below the offsprings. Okay. So I I will tell you. This is the normal symbols we are using. Now I will show you affected and normal. How can we recognize? It is very important. One question compulsory will appear in the NEET. You cross check in the NEET question papers. Compulsory will be having that. I'll tell you. We will say that autosomal dominating trait, autosomal recessive trait. Both the example I will show you. Very easily you can understand how you have to recognize. They will give you the condition. You have to write and you have to make it, and you have to confirm how many males affected, how many females affected, or you have to show that, isn't it? So I'll show you, for example, how it is representing. You can do so many. One from autosomal dominating trait I'll show you. One autosomal recessive trait I will show you. Okay. dystrophy. I'll show you. Autosomal autosomal recessive trait. Autosomal recessive trait and dominating trait there is there in that first I will give the example of autosomal dominant trait. Example for that myotonic dystrophy is it? Myotonic dystrophy is a disease. So how can you recognize which one is having a disease here? How can you recognize, dear students? So here we can see that what we call it as the condition in which normal, it is a dominating means always you should remember normal condition will be recessive. It is a autosomal dominating trait. If autosomal condition in that dominating is there, that one will be affected. For example, normal will be recessive. And what will be the affected one? Affected one that is in the heterozygous condition. If it is like this, it's affected. Now that is ऐसे देखो, that person will be having myotonic dystrophy. Okay, so how can we say if the marriage is taking place between a female, that is, if the marriage, that is a parental generation, parental one generation, if the marriage is taking place between the affected female with the normal male, what is the affected one will be having? Affected will be having, it is a dominant autosomal dominant trait beta. So it is capital S small a. What it will be normal? Small a. What is the possibility of their offsprings? The possibility of the offsprings we can see as it is not a sex linked. We can see both in the males also, females also. There is no chances of only female or male. We can see in the both beta. So if you see here, that is in the F1 generation. First, that is parental one generation. This filial one generation. In the filial one generation, okay, this is the F1 generation. That is the filial one generation of this parent. This parent's first generation you can see. So here you can see one male affected, one female affected. This is affected. Okay, and this is affected. So now, can you tell me what is the possibility? This remembering is very much important. Always you see, dear students, normal is small a, small a. Normal is small a, small a. Small a, small a. Small a, small a. Affected one are having, they are inheriting, they are getting the genes from their mother. Capital small a. Capital small a. Okay. If F2 generation also we can see here, F2 generation also we can see here, if the marriage is taking place between the normal and the infected, what is the possibility you can see? If there is a marriage taking place between these two. Okay, the next generation is relatives, closely related. If the marriage is taking place between these two, what is the possibility? If these two, we can see here there is a normal offspring to be there. Normal offspring to be there. Whatever male or female, because it is not sex linked. Sex linked disease है तो डिपेंड करता है फीमेल को जाएगा या मेल को जाएगा क्योंकि X linked है, Y linked है, मैं पढ़ाऊंगा आपको next में. So you can see here, this all we are able to see are the normal. But there you have seen, or else you can see here that is a female is affected or male is affected. At that time you can see that here. Can you tell me what will be the possibility here? The possibility is the one will be having one of the more significant. So can you tell me what is the possibility here? This is normal, this This is normal because none of the parents are showing any genetic defect. So all are normal. Here, one is affected because either one of the parent is affected. So it is also affected. So how can you recognize it? Because this is an autosomal dominant trait. So ये character कब appear होगा? The character is a disease. Okay, not actually it is a character. Genetic disease and character because of the presence of the dominating or recessive. That is autosome में तो autosome में अगर disease है, sorry, autosome पे अगर chromosome if it is autosome, the genes present on this chromosome we can call it as autosomal. If it is dominating, dominating. If it is recessive, recessive. Agar the chromosome is allosome, what we call it as it is a sex-linked disease. क्योंकि ये chromosome sex determinant होता है. So these are first to 22 pairs के जितने भी genes हैं, अगर उनमें defect हो रहा है तो क्या नाम देते हैं ना? We are calling it as autosomal. अगर dominating condition में disease हो रहा है तो उसको क्या बोलते हैं? Dominating trait. अगर रेसिसिव कंडीशन में हो रहा है तो उसको इसके जगह पे क्या करते हैं ऑटोसोमल रेसिसिव ट्रेट. One example I'll show you that one also. Okay, shall I write it? Now we will take an example that is autosomal recessive. Now, I will take the example of recessive gene. Did you all remember now? I have said it many times. This is dominating, this is recessive. Isn't it? So, shall I call it also dominating? Yeah, so also we can say dominating. If it is present both in the homozygous condition, then we can say. So, autosomal recessive trait. यानी कि ये जो डिसीज हम देख रहे हैं अगर रेसेसिव कंडीशन है तो ये एक्सप्रेस होता है नहीं तो नहीं होता है. सो दैट इज यू कैन से हियर एग्जांपल सिकल सेल एनीमिया. हैव यू हर्ड जस्ट नाउ आई सेड सिकल इसके बारे में पढ़े हैं जेनेटिक डिसऑर्डर्स में. आई विल एक्सप्लेन वेरी डिटेलली. सो सिकल सेल एनीमिया. So this anemic condition, the RBCs will be becoming sickle shape, unable to carry the oxygen properly. Okay. So this disease is because of the defective change in the condition and which will be expressed because of the recessive trait. अगर recessive condition में है तो ये disease express होता है. For example, इसके लिए हम देखेंगे कि जब normal है, can you people tell me what is a normal condition? Normal condition for this gene is normal कब होता है जब वो dominant है तो normal होता है. पिछली बार हमने क्या देखा था कि dominant है तो ही वो trait express हो रहा है disease आ रहा है. तो recessive का normal. तो हम normal कैसे देखते हो? In the normal condition how it will be done? Either capital A or capital A small. ये दोनों की कंडीशन में नॉर्मल रहता है. ये recessive trait है, ये disease है ना recessive condition में होता है. So when it is affected, when it is affected, dear students, it is affected when it is present in a recessive condition because it is a recessive trait. It is small a, small a. So let us see now if there is a marriage between normal. Let us see that is this is a male and that is a female. But it is showing one condition here. That is, the female is affected. What do you mean by affected? Affected means it is in the recessive condition. The male is either if it is like this also it is not affected or it is capital A also it is not. Okay. So here I can use this. Okay. So what will be the possibility of the offsprings beta? Possibility of the offspring, shall we check here? We can check here. Okay, so we are able to see here the possibilities of the. So now I will show the effective one. Just you try. Are you able to know how it will be? This shows the effective one. Okay. So if I draw the lines inside, you can say that it is an effective one. So effective one means this is depending on dominating है ऐसा नहीं कि confusion होता है दोनों के अंदर clear है so you can do any sort of problem because compulsory one question will appear in the image and each part from this. Okay, check it out later. In theory, not more than 2 marks. 1 mark completely definition. Usually they ask for 1 mark only. What is pedigree analysis? Sometimes 2 marks if they are asking, they are giving to write the symbols of the pedigree analysis. So here how can you say dear students? This is recessive. In recessive what can you the recessive. Remaining all we can say that is dominating. So what is they are not having this trait that is a type of disease. Okay. Now marriage between the what we call it as the condition also I will show you here. Here the condition is capital A, capital A. Okay, that is completely homozygous. Here also capital, for example, if it is also capital A, capital A. So you are able to see this is a both are normal. Both are normal. The carrier means it is having one small gene. Is it possible to generate if there is a marriage taking place between these two? Both are normal or not? But even though if I show you something, you will get surprised here. Both are normal. Isn't it? This is also normal, this is also capital A normal because this is totally dominating. In dominating condition, it is not showing. Capital is small a, and is A, capital is A is dominating over the small one. This is also normal. But if I draw the next generation, you will be very surprised here. So one will be changed. How can you say that one will be affected? How can you say that? Because they are getting one gene from the father, one gene from the mother. So there will be one will be carrier. One is female is carrier. Female is having carrier because she is getting one gene from the mother. So she will be having carrier. Please see in the male you don't find the condition. So here also I can say if I change here, you can also change here also. Okay. So first I have shown the normal condition. Now I am showing like this. So both are normal, but carry a small a is present now. So in one of the offspring, they will be showing homozygous recessive. So that offspring will be showing that this is expressing that trait to students. Same if you have to learn it. Okay, so it will be it may be like it is not showing effect, that is it is showing. Understood? It is about pedigree analysis. It is about the pedigree analysis. So we will stop here. We will continue in the next class, dear students. I hope you understood.